I'm Emily Hayes, senior writer with the Pink Sheet and Script. I'm here at the Biotech Showcase talking to Dan Gold, the CEO of MEI Pharma in San Diego. Hey, Dan.
Hi, thank you. Hi, how are you?
Good. So you were just saying this is something like your 16th year here at JP Morgan?
Yep.
Mm-hmm. Any initial thoughts about the meeting?
No, I think it's a good vibe. I think people are cautiously optimistic this year after last year. Yeah.
So your lead program is a drug for acute myeloid leukemia.
Correct.
We just had the ASH meeting in San Diego. There did seem to be a lot of development in the space. Can you comment about the surge in interest in
Yeah,
AML?
sure. I think that AML has always been of interest. You know, companies have been trying to crack that for a long time. It's just been very, very difficult. And I think now that we understand a lot more with the genetics, DNA sequencing, we have a much better sense. And there's just a resurgence of excitement around new compounds that may have an effect. And we're starting to see positive results. Of course, they're all phase 2. Nothing yet has been approved. But I think there is a resurgence in excitement around that field.
And what constitutes a good result in AML?
Well, the ones that we look for is actually survival. AML is an acute leukemia. That's the A of AML, and patients have a short horizon, especially in the group of patients that we are treating who are older. They are not really suited for high-intensity chemotherapy, which is really used for curative intent in younger patients or patients who are more fit. And in that case, where they're not fit for high induction chemotherapy, we really need new agents that will prolong their life. Responses, putting them into a remission doesn't seem to be sufficient to extend their lives. And so success is really measured by increasing survival.
And so you mentioned cytogenetics. We also heard talk at the ASH meeting about the many different subtypes in AML. So that's an opportunity, but it's also a challenge. How are you planning to address that in your development program?
Yeah, so you have sort of a segregation with some companies that are using targeted agents for some of these mutations. Unfortunately, in AML, there's no big subpopulation of patients that constitute a large fraction that you can go after with a targeted agent. It turns out that our agent, our HDAC inhibitor, when we combine it with azacitadine, seems to perform very well across many of these different subsets. The FLT3 mutations, the IDH mutations, they all seem to be susceptible to our therapy. So we are able to address a much larger patient population.
How do you explain the failures of the past? What do you think were the key factors?
You know, it's hard to know. I think it is probably multifactorial. Maybe there is some heterogeneity amongst the patient population. It's just a very difficult disease, as witnessed by the fact that there's never been an approved drug. To this day, there's really... In the US, that's correct. And to this day, there still isn't. Even for patients who are suitable for induction chemotherapy, we use high-intensity chemotherapies, but there's never been a formal approval of those drugs. And certainly in patients who can't take them, there's never been an approval. So it's just a very difficult disease. It might have something to do with the fact that there could be a stem cell component, and we're very good at wiping out the rapidly proliferating cells, but we aren't affecting the growth of those stem cells that rapidly then repopulate, and eventually the patient succumbs to the disease. The other problem is that this is a disease of the bone marrow, and so the patients themselves are very compromised. They don't clot properly. They don't make cells to fight fungal infections, bacterial infections. They don't make enough hemoglobin, or the cells that carry hemoglobin, so they're fatigued all the time. And all that just adds up to problems where they can die from symptoms of the disease instead of the disease itself. So it's a complicated question, but I think we are actually finally making inroads. Yeah.
Can you discuss the survival results that you've seen so far and what your plans are for phase 3?
Yeah, sure. So we presented data on 50 patients who were, the average age was about 76 years old. None of these patients would have qualified for induction chemotherapy for various reasons. Either they weren't suited because of their age, or they had other issues that would preclude them from getting intensive chemotherapy. And we used it in combination with a standard of care. It's called azacitadine, marketed by Celgene. Though not approved, but it's still marketed or sold in the United States and reimbursed by the insurance because it is the standard of care. Celgene had just run a large international study and showed that the average survival with patients in that category was about 9 months, 9 to 10 months, and what we showed is our patients were living on average about 19 months. So we almost doubled the survival of these patients, and for us that was very exciting. So now, based on that data, we've entered into a partnership with Helsinn Pharmaceuticals from Switzerland. They're going to take over the conduct of a large international phase 3 study, probably on the order of 500 patients. Should start that study very soon. And hopefully, if we can repeat what we've seen in the phase 2 studies, then that would be a major advancement for patients.
And when does that study read out?
We think it'll be somewhere from starting the trial to final data readout, about 2.5 to 3 years in that time frame.
Mm-hmm. And how much does a study of that size cost in AML?
Well, it's on the order of probably $80 to $100 million all told. Once you've accounted for the cost of the drug and everything else, it's not a small chunk of money. You know?
Would you like to talk about MEI 401, your candidate for B-cell malignancy?
Sure, I'd love to.
So that's a class that, can you talk a little bit about the class?
Sure, yeah, absolutely. So MEI 401 is a investigational drug. It's specific for a target that's called PI3 kinase, and it's selective for a particular form of called delta. Delta is very important in the survival of cells from the B lymphocyte lineage, non-Hodgkin's lymphoma, CLL, those type of tumor class. There is currently in the United States a single drug that is approved at PI3 kinase delta. It's called Zidelig or idelalisib from Gilead here in San Francisco, South City. As in many examples over years of drug development, new agents are very exciting, but then we start to learn some of their drawbacks. And this is unfortunately the case with idelalisib, or Zidelig, where it has very interesting activity in these patients, but it's fraught with certain side effects that make it somewhat problematic. Liver toxicities, cases of inflammation in the colon, in the lungs, and then after prolonged usage, immunosuppression, where patients are starting to see infections that are uncharacteristic for patients of this type. So I think this is one case where perhaps being next in line with a completely an agent that hits the same target but is unique in its structure and understanding a lot of the difficulties that Gilead has experienced, it gives us somewhat of an advantage as we think how to develop our drug. And that's what we're exploring right now.
And how many other companies are still pursuing the target despite the challenges that...
I think that there's probably a couple of others that have experimental agents, 2 or 3 others. We think our drug is excellent. We have presented a lot of data, preclinical data. We now have clinical data where we understand a lot about its action and its availability in patients and how it's working, and we're very excited. We think we have a top-notch drug. And
therapy in that area has been moving more toward combinations?
Exactly. Yeah, and so that's so important. I have to say, back to the question of the somewhat cloud over this whole field. I acquired this drug. I personally found this drug and thought it was really very exciting, but as in my role as the CEO, I always have to ask myself, I can't just fall in love with it just because it has to pass a lot of muster, and so we have to constantly ask ourselves, with the knowledge of Zidelig, is it in this cloud over the field? Will physicians use it if it in fact is good, or are we potentially going to spend a lot of resource in developing a drug that no one will ever use? And I think that in order obviously to get at that, one, we have to establish that it is worth using. But importantly, I personally went out and spent time with investigators, doctors who are actually treating patients, and they told me that this is absolutely critical to have a second generation that is safe, because they feel that this is such a critical target that offers patients a lot of benefit. And specifically for the question you asked about combining it, if they have an agent that has a much better safety profile, it would be much more amenable to combining with other agents in a safe manner, which they can't do currently because of the problems with idelalisib. So absolutely, cancer has always moved in the direction of combining useful drugs if they are not curative, and that's absolutely our intent as well.
And what stage is that?
So we're doing what's sort of considered an adaptive design. We're sort of a late phase one where we know the drug works, and we know that it hits the target, we know it's very active. So we're really not judging whether it has efficacy per se. We're really doing larger sample sizes of patients to judge if there's activity and if it's safe. So as soon as we establish that, then we'll immediately want to move into these combination studies. Yeah.
And just one final question, can you comment on your goals for the year, including financial and clinical?
Yeah, so fortunately with the partnership we did with Helsinn, we are in quite a good financial situation for a company of our size. We have certainly sufficient cash to see us easily through this year and well into the next year. Our goal is to help Helsinn get the phase 3 study started with our AML drug. We're also going to operationally run a second study with that same drug combination in myelodysplasia, which is a precursor to AML. So that's our goal, is to help Helsinn get started with the phase 3, get the phase 2 study in myelodysplasia started, and then read out our MEI 401 data and make the decision how we're going to go forward next. A lot of exciting things for the company.
Thank you, Dan.
Sure, thank you.